पेखे ओदी मैं पेखे ओदी ऊंचा मोहन सब ते
VIP लगे एने पढ़ लेने ਉਹਨੇ ਕਹਿਣਾ ਫਿਰ ਸਾਈਕਲ ਇੱਥੇ ਰੱਖ ਜਾ ਥਾਣੇ 'ਚ ਐਨੇ ਦਾ ਚਲਾਨ ਕਰਦਾ ਨਹੀਂ ਚਲਾਨ
Sorry class, good evening. I was a bit late. I went for cycling towards India Gate, but then I remember I have my class at 7 and I was in halfway, so I just made my route back. Sorry for the delay. It's 10 minutes, it's okay. I just wanted to know what we did yesterday, so for that I go to previous link. We were doing, I think, PCR or something like that we did. Oh. Yes, we did PCR. And we haven't done the DNA isolation yet. Cool. 10 minutes late. So yeah. Sorry, I just ran from my outside. Need just some time to put my mic and everything, get settled down with everything. Yeah, now I'm good. Fine, let's start. So you're done. So now I want to take you after the basics of your gel electrophoresis and PCR. Let's check about the macromolecules blotting. So like your DNA, big molecules like DNA, RNA. If you want to do blotting for them and want to identify which kind of sequence of DNA is present within the sample, so you go for first get the genomic DNA, you isolate that, then you digest them with the restriction endonuclease, and then after the digestion, you go for agarose gel electrophoresis, and after that results, that whatever that you get the bands, you need to transfer from the gel to the nitrocellulose membrane, because it's very hard to maintain the gel, and you cannot save the gel on the for the longer time, and your DNA could degrade from them. So in that's why the reason we use nitrocellulose membrane, and we transfer our sample from agarose gel, and after transferring it, we probe them with the phosphorylate 32P, which is a nitrocellulose membrane. Then we expose to X-ray film, and then after exposing, we got the results. Yeah, we got the results. So whichever the bands that we are looking for, we can get it. So in southern blotting, that was about no, like, so that was what just in general how the macromolecule blotting is done. And this blotting is of three types, that is southern blotting, northern blotting, and western blotting. The southern blotting, it's basically dealings with your DNA, and the northern deals with your RNA, whereas your western deals with your protein. So remember, southern DST, northern RNA NR, and western protein, so WP. So SD NR WP, like this way you can remember it. So in southern blotting, southern blot is a method for probing for the presence of specific DNA sequence within a DNA sample, and DNA samples are separated by gel electrophoresis and then transferred to a membrane by blotting via capillary action, and loss commonly used due to the capacity of other techniques such as PCR. And southern blotting are still used for some applications such as measuring transgenes copy number in transgenic mice, or in the engineering of the gene knockout embryonic stem cell line. Northern, it's done to identify the specific type of RNA molecule as a relative comparisons among a set of different samples of RNA. And RNA separated based on the size is then transferred to a membrane, then probe with a labeled complement of sequence of interest. And the intensity of these bands are related to the amount of the target RNA in the samples analyzed. So one of the most basic tools for determining at what time and under what conditions certain genes are expressed in the living tissues. So Western blotting. In Western blotting, proteins are first separated in size, yeah. Then in gel, they are sandwiched between two glass plates, and the technique is known as SDSPage, that is sodium dodecyl sulfate, and it's known as PAGE, polyacrylamide gel electrophoresis. The SDSPage full form is sodium dodecyl sulfate, yeah, sodium dodecyl sulfate. Then PAGE full form polyacrylamide gel electrophoresis. So this statement comes sometimes in the MCQs or in one word question that give us the full form. How is it? So the proteins in the gels are transferred to the nitrocellulose membrane like the same way like DNA and RNA, and which we probe them with the primary antibody, secondary antibody, and then over the secondary antibody we add the chemiluminescence, or we check either by the autoradiography. So they will detect the blots whether a protein is present or not. So these three techniques, southern, northern, western, uh, we will do them in details in our upcoming sessions. But today in this session, in the first sessions, I'm touching the summary of whole things that we want to study. Then comes your molecular markers that are based on the naturally occurring polymorphism in DNA sequence, that is your base pair deletion, substitution, addition, or patterns. The genetic markers are sequences of DNA which have been traced to specific locations on the chromosomes and associated with the particular traits. So it can be described as a variation that can be observed. So a genetic marker may be a short DNA sequence, such as sequence surrounding a single base pair change. A long one like mini satellites could work out for it. Then mitochondrial DNA, which is a forensic anthropologist which analyzes the hypervariable region of mitochondrial DNA to find the patterns connecting the victim to a relative. So you have nuclear DNA, mitochondria, nucleus. So all these are present, doing various like working data on that. So now let's check one by one. So these are the various genetic markers that are being used. RFLP, AFLP, RAPDs, VNTRs. So RFLP is the restriction fragment length polymorphism. AFLP is the amplified fragment length polymorphism. RAPDs are random amplifications of polymorphic DNA. VNTs are variable number tandem repeats. So all these come into this sequence, and SNP, STRs, SFP, RAD markers. So basically, that the most important ones are the first these sevens: RFLP, AFLP, RAPDs, VNTRs, SNP, and STRs. So each markers has its own importance, own level of working, and they help to make basically to your transgenic crops, to make your transgenic crops which are having a lot of transgenic traits such as drought tolerance, salt tolerance, heat tolerance, pesticide tolerance. So all these traits could be provided with the help of these markers. So the conditions that need to be in those markers, in these molecular markers, they should be polymorphic, codominantly inherited. Beyond that, they should be quite easy to use and cheap to detect. Data should be reproducible and randomly and frequently distributed throughout the genome. So molecular markers can be used for several different applications, including germplasm characterizations, genetic diagnostics, characterization of transformants, study of genome, organization and phylogenetic analysis, paternity testing, and investigation of crimes, which measures the genomic response to selection in livestock. Then RFLP, restriction fragment length polymorphism. This involves fragmenting a sample of DNA by restriction enzymes. which can recognize and cut DNA wherever a specific sequence is required. So a rare RFLP could be the length of detected fragment varies between individuals, can be used in genetic analysis. So in this, first we digest our DNA, our material with restriction enzyme, and then we recognize a specific part that is of our interest. And then after detecting the part on the DNA sequence that is of interest, we analyze it and look for the polymorphism. If it is quite useful for us, then we go for it. So that's why it's called as restriction fragment length polymorphism. Then AFLP, this is the amplified fragment length polymorphism. So in this case, we use PCR to amplify our typical DNA sequence after the digestion with the restriction enzymes. And then we go for the closely related species to identify the results. Then comes your RAPD. This is random amplifications of polymorphic DNA. So it is a type of PCR reactions, but the segments of DNA that are amplified randomly in that case. Then comes your microsatellites of polymorphism, SSRs or simple sequence repeats. So these are all are having the base pairs in which the sequences are repeated for 1 to 6 base pairs of DNA, and they are quite highly advantageous, like high variable, fast evolving, codominant, and relatively quite expensive, time consuming to develop. Then single nucleotide polymorphism, that is SNP, is a DNA sequence variation occurring when a single nucleotide, your ATCG, yeah, in the genome differs between the numbers of species paired chromosomes in an individual. They are used in the biomedical research crops and livestock breeding programs. So here we can see the single nucleotide polymorphism, that is a CG and AT. So AT could be snipped out from the CG, and we cannot, we cannot have the same results that we were looking for because of this short nucleotide polymorphism. And STRs, these are the short tandem repeats. In DNA occurs when the pattern 2 or more nucleotides are repeated and the sequences are directly adjacent to each other. The pattern can range in length from 2 to 16 base pairs. For example, is typically non-coding intron region, and they are used in forensic cases. And they're used for fingerprinting of individuals, genetic fingerprinting of individuals. Then principles of DNA isolation and purification. So DNA can be isolated from nucleated cells, and they are giant anion in solution. And the sources of the DNA that we can include are your blood, buccal cell, culture cells, bacteria, biopsies, yeah, forensic samples. So all these things we can use for the isolation of DNA. Further, for in routine procedure, if you want to do this routine isolation of a DNA for molecular analysis, so there are basic 3 steps in DNA isolation. First, you disrupt your cells, then you isolate the DNA out of it, then you precipitate that DNA. So that's the most common, whether you are using plant or whether using animal. So in all cases, we use the same method: cell disruption, isolation of DNA, and precipitating the DNA. That's it. So this is most commonly achieved by grinding, cell disruption by grinding, sonication, and removing the membranes. Whereas the isolation is done by removing the proteins by adding of protease, which will remove your all extra DNA, and precipitating the DNA using your isopropanol or ethanol. You use it could be used for isopropanol, and the DNA that we isolate is quite insoluble in these cases. It's not possible that DNA will soluble in the alcohol. So because of that, they will come and aggregate together, and we get a pellet afterwards, and we centrifuge it. So this step removes alcohol soluble salt. So you have cell lysis, protein degradation, then we bind them, then you do the washing, elute them, then you get the pure genomic DNA. Some basic rules while doing your DNA isolation. If you're using blood, you need to first lyse your cells, red blood cells, with gentle detergents such as Triton or X100, and then you wash your cells, which do not inhibit, like hemoglobin, which inhibits the friction enzymes and Taq polymerase. Then you work on the eye so that you can slow down your enzymatic processes, and wears gloves to protect your samples from us. And also use your autoclaves. All solutions that you're using, they should be stored in the fridge, except your SDS and organic solvents, and keep all pellets as supernatants until you have the DNA that you want. So getting to the DNA, so all cells in the present cells, so that DNA is stabilized and remains as a double helix. So which chelates magnesium is a cofactor of DNAs which chew up DNA rapidly. So anionic detergent SDS, which disrupts the lipid layers to dissolve membranes and binds positive charges of chromosomal proteins to release the DNA into the solution, which includes a protease to digest the proteins, incubate the solution at elevated temperature for 4 to 24 hours. So getting rid of the protein, organic solvent extractions could be used using equal volume phenol into chloroform, that is 24:1. Then protein at the interference, after centrifugation, use 10,000 rpm at 10 degrees Celsius for 10 minutes. Then you precipitate the DNA with the help of your ethanol, 95% ethanol, ice cold. We add around 2.5 to 3 volume of our sample of DNA and leave over the night at minus 20 degrees Celsius. Then we centrifuge at 10,000 rpm for 10 minutes, 4 degrees Celsius. Then you wash this DNA pellet, remove the excess salt in ethyl hydroxide and air dry. Then you resuspend in the sterile distilled water, that is pH 7.4. Then store at 4 degrees Celsius, or froze your DNA at minus 20 degrees Celsius for the long term. So that's the you remove your protein, you get your DNA with these materials, maintain some basic rules while in the lab, and these are the basic principle in order to isolate your DNA. Then quantify the DNA. So to quantify, the formula that we use for DNA concentration is microgram per ml. OD 260, 100, dilution factor 50 microgram per ml, 1000. So nucleic acids have peak absorbance in the ultraviolet range at about 260 nanometer, and the 260 units OD units that we use is our double stranded DNA, single stranded DNA, and RNA. So for the 260, it's 50 microgram per ml. For 260, it's 33 microgram per ml. For 260, it's 40 microgram per ml, like so. Then to check the purity of your DNA, you check the ratio for the two different spectrometry units, that is 260 and 280. If the ratio is less than 1.6, then it's protein contaminated. There is a lot of protein in your DNA sample. If it is more than 2, then it's a chloroform or phenol contaminated. That is, you have a lot of ethanol particles in your sample. So we have to be taken care that we don't have much of the protein and also not much of the ethanol. Both are removed while at the end of our DNA isolation. Then you repurify the sample. So in summary, in this DNA isolation, we have discussed how to make your sample for DNA extractions, lysing of the cells at elevated temperature. We use detergents and enzymes in the salt buffer. Then we remove the cellular proteins. Then we precipitate the nucleic acid with ethanol. Then at the end, we quantitate and purity of measurement of the DNA at the end of the case. So future aspects: agriculture development and environmental protection to ensure food security for ever-growing human population. So that's it. That was about DNA isolation, how DNA isolation is done. Now we go for a quick DNA isolation video.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipetters are on the left, racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotech collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter. Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. Prior to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. Tubes containing 20 microliters of DNA Genotech's proprietary purifying solution, Prep It, will be provided. Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution. You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex. To turn on the vortex, press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration. Repeat this process for all remaining samples. Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward. And balance the tubes across the rotor by ensuring that all tubes have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case, 5 minutes. Press the arrows by the speed to navigate to the proper RPM. In this case, 14.5 thousand RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that there are no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately. As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube. Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples. Precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample and mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes. Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA. Visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process. Using a P1000, slowly remove and discard approximately 800 microliters of supernatant. Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples. Visually inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample. Do not mix this solution as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision. This will take 2 or more draws. On the first draw, you can expel the solution into the discard bucket and use the same tip to draw up again, as long as the tip has not touched other surfaces. Repeat this process for the remaining samples. Lay out a few layers of Kim wipes. Open the tube and knock it onto the Kim wipe to remove any large volumes of solution still remaining. Leave the tube open and lay it on its side to evaporate any of the remaining solution from the pellet. Repeat this process for all remaining samples and allow them to air dry for 20 to 30 minutes. If the tube still has spots of solution or if the pellet does not look dry, allow for evaporation to continue. If the pellet looks dry, then resuspend the DNA using water. Add 100 microliters of molecular biology grade water directly onto the pellet side. Cap the tube and mix by vortexing for a few minutes. Visually inspect the tube to ensure that the pellet has disappeared or has gone back into solution. If it has not, then continue to vortex or allow the tube to sit at room temperature before proceeding to subsequent steps. Repeat this for all remaining samples.
So that was about DNA isolation. part 1. So you can watch the part 2 corresponding to that later, I guess. Yes. So that's it. I think I had shared this presentation with you already. So we are done with DNA, PCR, uh, your macro blotting, and various other techniques we have discussed in today, various molecular markers, the codominance, and all these things we have discussed today very well. So I can't, if I start another topic, it will take me whole half an hour. So we can make the next topic for tomorrow start. Tomorrow we can start that, and yeah, we can initiate that part tomorrow. So that's it for today. Thank you very much. Codominance in case of RFLP, but Sanjukta ma'am, I think it's because of the um, because this polymorphism in this random restriction fragment length polymorphism. So whenever this is happening, so it's making the results that you're getting, your genes, that your markers that you're getting, they're codominant always. Yeah, that's why it could be advantage. So Rahul, yes, this protocol in every lab, because all these protocols are are different according to person. Because when I started to work in my lab and want to precipitate some DNA in order to find out some results, for my redox biology research. So at the end, what happened? I was doing these experiments on daily basis, and I was getting the results as the way I want. So the way I change the protocol, you get the hold on the protocol so much that when you add 1 microliter of something, you get something else. When you add 2 microliter of something, then you get something else. So things get changes according to it. So it changes every time. No, air drying does not contaminate. You just keep it in the air. You can make it some, you can put in the spinning column and close it by the glasswares, but it does not contaminate much. It's okay. We used to put our samples in the air forever. Our gels used to be in the water, in the buffer for like 1 year or so. So nothing happened to them. We can still using them sometimes. So Shivam, these steps uh, they are not so difficult actually. So even when I started to do all these steps, these practicals, what you do is you write them on a paper. You take a white paper, you write step 1. This is step 2. This is how the protocol things are going on. A very good question actually. So when we started to work on the protocols, so you have to be careful that you should know what you are doing. And because for some people, what happened, everything is in their brain, so they don't need to write anything. For some, it's like they need everything to be written in front of them, then they could start the experiment. So I am the second one. Unfortunately, I need to write everything before I go to the lab. So these things I have to do, because while doing these experiments, a person tend to do mistakes. It's normal. It's very normal to do the mistakes. And some people, even though they do the mistake, they don't admit it, that I did the mistake. Because if we started to admit the mistakes, then people will think that we are not so good in our work. But if we admit once and we do not do that mistake again next time, that's the thing we have to learn in the research. That it should not be repeated one more time. Admit that we did that something wrong, and from the next time things will be fine. So you just write everything on a white paper, then keep reading what is the next step, then do that, then read it, what is the next step, then do it. So at the first time when you were doing, you will do a lot of mistakes, a lot of things will be, results will be might be not coming. But By the third time you do the repeat thing, by the in a week, if you do 5 times a week the same thing, the sixth time you don't need that paper. Your hands will be doing everything automatically. So that's eventually I learned science. By the end of my work, I was just everything started to be in my brain, and it's just my hands are just working automatically, and we don't need any protocol or something. We just working as our daily routine, getting the PCR done, making Western blot, going for, getting DNA microarray, giving our sample to that lab, and then side by side, you have to write your papers, you have to write your publications, you have to write for your grant, So being academia, it's like you are having 10, 5 heads, 10 hands, and 20 legs maybe, and everyone and everything is working very nicely and in systematic way. So sometimes this happens. So that's something funny part of being scientist. And now next, there are even some dominant markers, right? Yes, it's they are also dominant, co-dominant, both are present. So Anjali Mohole, so to learn these in practical. So I mean, I haven't. This is you can learn from here. Actually, you're most welcome to learn from me. This is what this workshop is for. This molecular biology and biochemistry technique. So we eventually will learn a lot of techniques in a virtual way, unfortunately, as we having this COVID time. But to do practical way, there are some workshops going on where they're giving internship pattern like training for like 6 weeks, 7 weeks. So you can approach some companies, or you can apply at some big research institutes, CSIR labs, or IITs. Somewhere you could get a research position, and you can do all these research works on your own. No, I mean, they are blunt ends, they are sticky ends. So according to what kind of friction enzyme, with which kind of friction enzyme you want to cut, so it depend on that. Yeah. So you have to see, um, like what are you looking for, which segment you want. So according to that, because each friction enzyme has its own different role. They are cutting at different sites, cutting different series of your DNA segments. So it's different from place to place. Yes, Laiva, I agree. But most likely we will be done with the lectures around 4th of May or 5th of May. Oh, sorry, June. So that's why I also have extended the date for the review article. You still all have 1 month. So don't worry. You all have ample amount of time. Wake up 6 in the morning and you sleep at 10 in the night. So you have more than 12 hours, or not even more than, you have 18 hours or so in a day to work. So if you have 1 month for 18 hours, so see, you can multiply. How much is it? 18 hours by 30, or 25, 24 days. Oh, you have 3,456 hours. I think that's quite ample time to write the review article. Yeah. So it's a lot of hours are there. So make sure your all hours are being used. Just being funny. Anyhow, uh, yes, we can extend to be on serious note. That no worries. But problem is, if you send your review article late, what will happen? You will get your certificate late also. So the more earlier you send your review article, the earlier you will get your training letter also. So they will be both proportional to each other. So any further questions, bachcho? Students, any further questions? Yeah, by 7th of June, yes. It's a lot of days. The thing is, I have no problem. You can extend it. The things will be better. But we were told that when the internship is over, when the our lectures are getting over, by the end of that, you need to submit it, because for us the next batch will start. Our new things will start, so it will be not possible to entertain the previous old students. For us, it's difficult actually. So that's why I keep this time frame. Uh, and in the time pressure we can work actually if we keep make things done extended. For some students who have genuine problems in the house, as the time is not going on, even the last batch of our April May I have extended for some students due to some genuine reason, which I cannot share here. It's horrible time actually we are having at the moment, but yes, we can extend it, don't worry. But for our authorities it will be difficult to give you the letters then. So that's why I'm saying to keep it around 7th of June or so. So okay class, then we see us tomorrow, same time, same place. We will tomorrow learn about cell culture techniques. Thank you, bye bye, take care, stay safe.